This is Sujata Bembade from MSc Biotechnology first year, and my topic is cancer immunology. Cancer immunology is an interdisciplinary branch of biology concerned with the role of immune system in the progression and the development of cancer. The most well-known application of cancer immunotherapy is cancer immunotherapy, where the immune system is used to treat the cancer. The cancer immunosurveillance is a theory formulated by Bernard and Thomas in 1957, which proposed that lymphocytes act as sentinels in recognizing and eliminating continuously rising transformed cells. The cancer immunosurveillance appears to be the most important host protection process that decreases cancer rate through inhibition of carcinogenesis and maintaining on regular cellular homeostasis. It has also been suggested that immunosurveillance primarily functions as a component of the more general process of cancer editing. Here is the image which shows tumor-associated immune cells in tumor microenvironment of breast cancer model, which shows various type of tumor-associated immune cells. Next is tumor antigens. Tumor may express tumor antigens that are recognized by the immune system and may induce an immune response. These tumor antigens are either tumor specific antigens or tumor associated antigens. Tumor specific antigens are antigens that occur only in tumor cells and can be the products of oncoviruses like E6 or E7 proteins of human papillomavirus occurring in corneal carcinoma, or EBNA1 protein of EBV occurring in Burkitt's lymphoma cells. Another example of tumor specific antigen are abnormal products of mutated oncogenes or anti-oncogenes. Next is tumor associated antigen. Tumor associated antigens are present in healthy cells, but for some reason they are also occur in tumor cells. However, they differ in quantity, place and time period of expression. Oncofetal antigens are tumor associated antigens expressed by embryonic cells and by tumor. For example, oncofetal antigens are alpha fetoproteins produced by hepatocellular carcinoma, or carcinoma embryonic antigens occurring in ovarian or colon cancer. More tumor associated antigens are HER2, EGFR and MAGE1. Immunoediting. Cancer immunoediting is a process in which immune system interacts with tumor cells. It consists of three phases: eliminating, equilibrium and escape. These phases are often referred as the three A's of cancer immunoediting. Both adaptive and innate immune system participates in immunoediting. In the elimination phase, the immune response leads to the destruction of tumor cells and therefore the tumor suppression. However, some tumor cells may gain more mutation, change their characteristics and evade the immune system. These cells might enter the equilibrium phase, in which the immune system does not recognize all tumor cells, but at the same time the tumor does not grow. This condition may lead to the phase of escape, in which the tumor gains dominance over the immune system and starts growing and establishing immunorepressive environment. As a consequence of immunoediting, tumor cell clones less responsive to the immune system gain dominance in the tumor through time as the recognized cells are eliminated. This process may be considered akin to Darwin's evolution, where cells containing pro-oncogenic or immunosuppressive mutation survive to pass on to their mutated mutations to their daughter cells, which may themselves mutate and undergo further selective pressure. This results in tumor consisting of cells with decreased immunogenicity and can hardly be eliminated. This phenomenon was proven to happen as a result of immunotherapies of cancer patients. Next is tumor evasion mechanism. CD8 plus cytotoxin T cells are the fundamental element of antitumor immunity. Their TCR receptors recognize antigen presented by MHC class 1, and when bound, the TC cells trigger its cytotoxin activity. MHC1 are present on the surface of the all nucleated cells. However, some cancer cells lower their MHC1 expression and avoid being detected by the cytotoxin T cells. This can be done by mutation of MHC1 gene or by lowering the sensitivity of an INF sensitivity to IFNY. Tumor cells also have defects in antigen presentation pathway, which leads into the downregulation of tumor antigen presentation. The defects are, for example, in transportation associated with antigen progression or tapacin. On the other hand, a complete loss of MHC1 is triggered for NK cells. Tumor cells therefore maintain the low expression of MHC1. Another way to escape cytotoxin T cells is to stop expressing molecules essential for co-stimulation of cytotoxin T cells such as CD80 or CD86. Tumor microenvironment. The production of TGF beta by tumor cells and other cells leads to conversion of CD4 plus T cells into suppressive regulatory T cells by the constant contact development or independent stimulation in the healthy tissue. Functioning tracts are essential to maintain self-tolerance. In the tumor, however, tracts form an immunosuppressive microenvironment. Tumor cells produce specific cytokines to produce myeloid derived suppressive cells. These cells are heterogeneous collection of cell types including precursor of dendritic cells, monocytes and neutrophils. MDSC has suppressive effect on T lymphocyte, dendrite cells and macrophages. They produce immunorepressive TGF beta and IL-10. Another producer of suppressive TGF beta and IL-10 are tumor associated macrophages. These macrophages have mostly phenotype of alternatively activated M2 macrophages. This activation is promoted by Th type 2 cytokines. Their effect are immunosuppression, promotion of tumor growth and angiogenesis. Tumor cells have non-essential MHC class 1 and on their surface, for example HLA-G and HLA-G is inducer of tracts MDSC and polarized macrophages into the alternatively activated M2 and has other immunosuppressive effect on the immune cells. Next is immunomodulation method. Immune system is the key player in fighting cancer. As described above, in the mechanism of tumor evasion. The tumor cells are modulating the immune response in their profit. This is possible to improve the immune response in order to boost the immunity against the tumor cell. Monoclonal anti-CTLA4 and anti-PD1 antibodies are called immune checkpoint inhibitors. Thank you.